Welcome, everybody. So today we've got Paddy Royall from ESPCI in Paris, and he's going to tell us about 3D active matter, colloids to fish.
Okay, thanks everyone for coming. So I think maybe the first thing to say is that I'm coming from an experimental background, and if there's anything that doesn't make sense or wants further clarification, things that I would gloss over because I wasn't thinking about it, then do please stop. I'm more than happy to explain whatever it is. Okay, so I'm going to talk about colloids, and then moving on to fish towards the end of the talk. The people involved, you'll see our illustrious. Okay, so this is what I mean by it's my sort of standard picture of colloids. And the idea here is that we increase the packing fraction and these things organize into crystals. So they've got the same phase behavior as a mystic system would have. They're much, much simpler. These particular ones are just spheres, which is more or less the simplest of lattice material that one can think of. But what we can do with them in particular, why they're useful for our purposes, is that what I'm interested in is being able to take pictures of them in 3 dimensions. So what I hope you can see is these red dots here are these red dots are the coordinates that we track the particles. This is a 2-dimensional slice of a 3-dimensional image. And clearly I'm on a 2D screen, so I can only show you a 2D slice, but the data is 3-dimensional. What we get are 3-dimensional coordinates, as if we had a computer simulation of this. Well, this is an experiment, and these things are self-assembled into a base of crystal. Now, clearly, a lot of things do that. A lot of molecular things do that. So you might say, well, why do we care about the colloids? We can study them where their interactions are simple. They're spherically symmetric, which is like this one, in this case, and we have the pair correlation function between them. And what we can do is very accurately reproduce what they do with a computer simulation. The lines here are computer simulations written with a very simple Monte Carlo code, with this very simple interaction between them. And we can use the Einstein-Zerner correlation to argue that if we know the pair correlation function, then we know the interaction potential. If we know the interaction potential, we know what the system is doing. We know what state point it can crystallize. We can predict this with very high accuracy. That in itself is all fine, but you could do that with a molecular system if one could do it well enough with scattering. But what is very hard to do, unless you can actually see the thing in real space, is higher order correlations. So these are higher order correlations. This previous thing I showed, which is pair correlation, these higher order correlations can be predicted. So the solid lines are predictions from an approximation of density function theory, and the small data points are computer simulation, and the bigger data points are experiments. And we can predict those these higher order structures, or rather complex structures, up to 13 particles in this. And we can then predict what these higher order structures are important in certain phenomena like the glass transition, crystallization, and this is the kind of thing we really can only access or access by taking real space measurements. So the first thing that I want to talk to you about, that was all passive. Okay, that was sort of background. That's what we keep taking with the method. What I want to talk to you about is some active colloids. And the first system I'll talk to you about, I will go into 3 dimensions later, but these are the Quincke Rollers. And they're active, they're driven by an electric field. I'll say a little bit more about them in a moment. But what you see here is that they have undergone something that looks like microphase separation. That is to say, they have not formed a large bulk liquid, but they're clearly not homogeneously distributed. This is the same sort of data. What we've done here is to track the particles, and they're colored according to a bond orientation order parameter, psi6. What that means is that if it's pale blue, they're hexagonally ordered, and the redder it is, the less hexagonally ordered. So this is experimental data. We track the coordinates, and we can analyze the structure dynamics. So that's to introduce the system. what actually happens, the Quincke rollers in the context of active matter were originally by Denny Bartolo about a decade ago now. And the idea is to have a particle in a dielectric fluid, we apply an electric field, particle polarizes, and above a critical field strength, then we have that the particle is starting to rotate. Okay, so then if we apply enough field, the particle rolls, and it's intrinsically then a two-dimensional system because the particle is on the substrate, and they are rolling in some direction. Now, this might be a little bit uncomfortable, but not all Quincke rollers are the same. And were I to be further west in Bristol, I might use the relevant accent, but they make them differently in Bristol. And what happens? So what happens is that this thing's an electric field, and there's a solvent. Solvent flows along the plane, and then in practice, what we have above the particle is a lot of space. So the solvent can move along the plane, it dumps the particle, it then moves up. And well, that's all fine if I have a single particle, and it will still do the Quincke roll in itself. But if I have two particles, then they start having an attraction. Okay, the solvent flowing on either side of the two particles, but not so much between them. That leads to a long-range electro-hydrodynamic attraction. This only happens in Bristol. I don't know why, but well, I can discuss why, but for our purposes, it happens in Bristol. There are other examples of Quincke rollers in literature where it doesn't happen. It doesn't happen anything like as much. But this does mean that the particles behave differently. And we've added a long-range attraction term into their equations of motion, such that we can describe this long-range attraction that leads to them, leads to this behavior. So in particular, what we have as we start to turn up the field strength is that we have a they're just passive grounding particles at low field strength. At weak field strength, I said that there was a critical field that makes them active. Below that critical field, the electrohydrodynamic attraction is still there, so they crystallize like this. But at some point they become active, we then call them amoeba, and when we make them even more active, these amoeba essentially vaporize, and we have what we call an active gas. So that's the beginnings of phase behavior. Let me show you what it looks like at the particle level. These are what we're doing is turning up the field here. So we start with a crystal, and eventually it becomes an amoeba, and then it evaporates because we're increasing the field, increasing the activity throughout this process. Now, we've implemented the model that Denny Bartolo built in his original paper with our additional long-range attraction, and we can map the parameters accurately between the experiments and the simulations. And if you look at this, you'd say, okay, it's doing a reasonable job of reproducing the experiments. What you see in the experiment that's pretty similar to what you see in the simulation. I'm going to claim, and you may agree or not, but I think there's more kind of turning around going on in the experiments than in the simulations. And I'm happy to discuss that further, but at least the simulations are doing a pretty good job of reproducing this. We can, this is now, I have to be honest with you, I'm not sure if this is experiment or simulation, it doesn't matter. This is what happens at very high field strength. At high field strength, the system on field band. The blue again is showing that the blue is high rooted to high 6, high bond order, digital order, and high density. So the system's undergone again a sort of really big density fluctuations due to driven this time by activity. This had previously been seen by the work by Denny Bartolo, but what they hadn't seen was the lower phase separation at lower density levels. So let me just go back. This is now zooming out of a simulation in this very high field strength phase where we're seeing banding. And so what we have is something which is a little unusual. We've got two mesophases in the system. That is to say, at low activity, we have these amoeba. What's happening with the amoeba is that the long-range attractions between particles encourages them to phase separate here, but they don't fully phase separate, they just form these domains within the size. But that's opposed by the activity. to prevent them completely phase separating. However, here, high activity, then what we find is it's actually the activity that drives the phase separation. Okay, it's a little bit reminiscent of NIPS, at least in the sense that activity is driving it, but it's not the same thing. These things have orientation, alignment correlations, interaction between them, which is not required for NIPS. Okay, so there's an analogy here between what we see with this active matter system and what one often encounters in various soft materials. In particular, this formation of these mesophases. These are dipoc polymers. These are examples of the kind of interaction you typically need is something with a short range of traction and a long range of repulsion. Okay, the short range of traction drives the system together. The longer range of repulsion stops it condensing into a single phase. So we've seen these sort of mesophases before. What we've not seen is them being driven by activity and suppressed by activity in the same system. In the end, we arrive at a phase diagram as we show here. Now, what I quite like about this is there's a lot of data points on it. Some of them are experiment, some of them are simulation, and I don't know which are which because the group is that good. Okay, so we can completely reproduce these phases that we see. Mainly I've talked about this side of things where the A is the lever, and then we've got this banding phase that feels and the crystal that. So that's the sort of bulk phase behaviour of the crystal Quincke rollers. What I now want to do is talk about how they behave in confinement. So what we can do, they exist in an electric field. So what this is some confined Quincke rollers. They're not physically confined, they're not in little pits. What they're in is regions where the electric field is on and regions where it's not. Okay, so in these are cylindrical regions where we apply the electric field over them. And what you can see is that they have there's different numbers of particles in each cylindrical. So we can study then the behavior of this system as a function of the number of particles. Now it turns out that for the size of the pits that we have, if we have 71 particles, that makes a perfect hexagon. And so that turns out to be the largest number of particles we can have. Because the pits themselves they are circular, the field is roughly circular, but it turns out that the best pattern is hexagonal because we don't have a physical circular band. So this hexagonal pattern is effectively the densest pattern we talk about. So when I talk about N max up here, this is very, very dense pattern. And they become more and more hexagonal as we go to higher packing. I also want to introduce this polarization order parameter. The polarization measures the extent to which the particle can move in the same direction. So this is the particle velocity, and the polarization is 1 when they're moving together, and 0 when they're random. What I want to focus on is the high density. Okay, so there are a number of, we've coloured some state points here. So yellow is a weak field, then red, blue, and then green up here at the high density. So I will come back to those colours in a moment, but let me show you the movies of what happens to this system as we increase the fields. When it's just become active, the system's a rigid rotor. The colours here, colours mean that just so that we remember which particle is which. Okay, they don't mean that all the particles are identical. This is simply the remembering the history. If we start to increase the field strength, then what we see is that we're starting to get some slip. Okay, so the particles in the very outside layer here are not moving as fast as the ones in the layer in between. And in fact, the layer in between appears to be moving faster than the layers inside. Those layers inside. Now we can turn the field up some more, and what we see is that, oops, Right. So again, there's more stuff going on, and we've now got the outer two layers are slower than the interior. And then if we turn things up even more, we've broken the hexagonal structure, and we've also got the situation that the outer layers appear to be going in the direction to the interior. Okay, so we can capture this behavior, perhaps surprisingly, with a discretized tube model. So we've clearly got a very small system, there are 5 layers in it, and we discretized the tube model to describe that with appropriate boundary conditions. That's what the lines are here, and the claim is that it qualitatively captures the inversion behavior at a high field strength. The interior is moving very quickly, low field strength, the exterior part moves fast. So that's something about how they behave when we combine them, but we can also talk about clusters or molecules, if you like, the Quincke Rollers. There was quite a lot of interest about a decade ago making colloidal molecules. How did groups of colloids behave differently to individual particles? And so we thought we'd have a look at that in the Quincke Rollers. This is a dumbbell. It's sort of possibly the simplest kind of dumbbell Quincke Roller molecule that one could come up with. And what you see is that rather than going in a straight line, it's going around in circles. And one might say, okay, that's fine. I mean, because I've got 2 spheres, they're rotating. No colloids the same size have ever been made. So they're bound to go around in a circle because they're rolling, yet they're different sizes. So they have to go around in a circle. So that's fine. That's all straightforward. Except, because of this bit, at low field strength, the centre of mass doesn't move. So what's shown here is this is the centre of mass. So it's not moving. These are the tracks of the centres of the particles. So they spin at low field strength. And then at a higher field strength, they start to do the orbital motion that I showed you. And then at an even higher field strength, they go around in a circle, in a slightly elliptical orbit, but not so far from a circle. But there's none of the disordered behavior at lower field strength. But then the even stranger is that all the circles have the same radius. Now, if the colloids are colloids, and I said that it's obvious that they'll go around in a circle because they're different sizes, but they'd have to be different sizes in the same way if they were going to go around with the same radius. So that's not what's happening, because they're never going to be different sized in the same way. You might have a very big one, a very small one, 2 almost the same size. If they're all the same size, the radius huge. If there's a big one and small one, the radius should be tiny. So why is the radius always the same size? So it's clearly not the fact that they're different size. So I mentioned that if we have a monomer, then there's solvent flow around the monomer. And if we then have a dumbbell, well, the solvent's still going to flow around the dumbbell. And the picture that we have, which may be naive, but picture if we look down on it, there's mass coming into the dumbbell in the form of the solvents coming in and then flowing up around it. And it's a bit like an accretion disk in the form of the solar system, in the fact that if there's mass moving into something, if there's any angular momentum in the mass, that will be transmitted to the system, and so so we think that's why they're spinning. But what that entirely does not explain is why there's a transition for something which is totally disordered like this to something which is ordered in terms of the field. Okay, but it does explain why they're going around the same way. The next thing one might want to ask is, well, what happens when dumbbells meet? Okay, so 2 of them can collide with one another, and then they're going to be stuck. Well, it turns out that under certain circumstances, if they are soft enough, so if they're hard, they always can get around each other. But if they are soft and they're driven with the right amount of speed, there's a region here where they are stable. So these will form a boundary plate But there's some distribution of the lifetimes, even in the unstable region, they'll form a bound state for some time. Because even if they're hard, down here they can form a bound state for some considerable period of time. This distribution of lifetimes that they form the bound state for. And of course, the standard of doing experiments is that one never really knows if the thing's in a long-lived metastable state or if it's just in a stable state. But anyway, this is what they do. So here's an example of two dumbbells that hit each other, and then they're in the bottom left of the screen, bottom right of the screen, two dumbbells collide and come together and form what we're calling a tetramer. So I like to think of these things as a hydrodynamic version of the electric bonding between atoms to form molecules. These things are instead bound by hydrogens. We can go one stage further and have a dumbbell hit a tetramer, and that will then form what we call a hexamer. These seem to be a very excited state. They spin very quickly, and they're also stable. But this I'm claiming is a succession of states which the system can form by being bound together to interactions. I should mention the work we did to study them with computer simulation included hydrodynamic interactions through multi-particle collision dynamics. We can put three particles together, which is what we've done here. And if we have three particles, then they can't roll because a triangle sitting on the substrate cannot roll. What it does instead is to flip. And hopefully you can see that it's exhibiting a flipping motion. In the on the lower left of the screen, then we have on the lower left of the screen, then we have this honeycomb lattice. What this is, the center of mass of the trimer carves out a honeycomb lattice because when it flips either through moving through an angle of 0 degrees or this here, so this enables it to trace out the honeycomb lattice plus diffusion, because the thing is, of course, colloidal and it's exhibiting translational diffusion as well, but it roughly measures out. Okay, so that was what I wanted to tell you about the Quincke Rollers in 2D. And but what I really wanted to do when active matter always got going was to make a 3D system. So what we did was to consider what systems might be available that we could take into 3 dimensions. And the best thing we could find, or best thing we marketed, was this induced dipole system. So let me tell you about the particles. One side, this side, is coated in metal. But crucially, it's then covered in the same material, which is non-conducting, that the rest of the particles are made. That's essential, because otherwise they will stick together irreversibly density touch each other. So we cover this metal thing with a thin layer, 30 nanometers of iron silicon starting. What the particles do, well, we put them in an AC electric field, whose frequency we can change, but today I'll talk about relatively low frequencies of 5 kilohertz. And that does 2 things. It makes them move in the XY plane, so they are a little bit like active Brownian particles in the XY plane, but they also have a dipolar interaction. We know generally that putting colloids, if there's any ions around, which there usually are, in an AC field, this will induce dipolar interaction. They will then attract each other along the z-axis due to the dipolar interaction, and they'll repel. Okay, so we know what the dipolar interactions will do to the system extensively from previous work with passive systems, and this will give us a guide as to what we might expect. Essentially, we're taking this passive system and adding activity to it in the XY. So the passive system has a rather complex behavior. As we increase the volume fraction up here, we go fluid to body center cubic, base center cubic. There's more body center cubic here. There's body center tragonal, and something called string fluid. And this is string fluid to tragonal coexistence. It's a complex system making a number of crystal phases, and it even makes fluid phase, this thing where the particles organize into strings. So that's what the passive system does. What we're going to do, as I say, is add activity to it in the XY plane. So if we look at our active string, this is what we see at a low volume fraction, and then what we see in the microscope. This is now a rendering. It's an attempt to capture a three-dimensional image in 2D. I'm not that convinced of it, but anyway, this is what we've got. So what I hope you can see are some columns moving around, which are moving around an awful lot more than they would do from their ground location. This is the active strings, and so what's being done here is the field strength is increased throughout the movie, so that initially they're not moving very much, but then they start to move more. Okay, so at higher volume fractions, the strings percolate to form sheets. This is a new structure. Okay, so it has been seen, it can be seen transiently in the passive system, but this is steady state. And what we're doing here is we're looking down on it in the XY plane, and this is the sign. So this structure isn't present as far as I know, not otherwise available in any kind of long-lived way. If we increase the volume fraction, we see that this thing percolates in 2 dimensions. So there's a 2-dimensional population transition here, and at higher volume fraction, they form the crystal, body center tetramer crystal, passive system, and at high volume fraction, still they form an hexagonal crystal, which is what spheres generally do if we get them to a sufficient density. Okay, so we've got this complicated phase diagram of this passive system. What I emphasize is that these sheet phase and this labyrinth phase, these are not available otherwise. Okay, so this is the only system I know that can produce these states. We have captured it in computer simulation. Let me see if the movie will be working. Okay, so this is computer simulation. This is exactly what I said. It's Brownian dynamics with active Brownian particles in the XY plane. We're looking down on it. This is the sheet phase, and the dipoles that you see are related to the fact that, or a representation that the one side of the particle is metal and one side is not, and that's what drives the motion. What you see here is one of these traveling colloids. The dynamics are peculiar in this system. One usually imagines that the relaxation time in a colloidal system increases as we increase the volume fraction, but this was non-monotonic. So particularly here, at higher field strengths, there is an increase in the relaxation time, and it drops when it crystallizes, which is just strange, to say the least, and then it continues to increase at higher volume fractions. The labyrinth phase is a percolating gel-like phase, and yet it's going faster. That's slower. But then when we actually crystallize on the crystal, then it persistently accelerates, and this seems to be most strikingly unusual. Okay, so that was roughly what I wanted to tell you about the active colloids in 3 dimensions, and now I'd like to move to fish. So we're coming at the fish from the background of tracking things in real space in 3 dimensions. That's what we'll do with the fish, which are obviously much larger than colloids, but that doesn't make them easier to track, it turns out. Mainly because the colloids, what I should have told you, the 3-dimensional colloids, the refractive index of the colloids is matched with solvent, so the whole system's transparent, and that's not the case for fish. Fish are a little bit translucent, but they're certainly not transparent. So instead of the system with 3 cameras, we have a parabolic shaped bulb, a parabolic shaped observation tank. The fish do not live in this tank, all right? The fish live in a normal fish tank, okay? But we put them into this tank to make observations. We also use a relatively large tank. In that the fish are, liver fish are about the size of the top of a head, okay? So they're not very big. But initially we had a smaller tank, and the fish went to the edge. And we thought, okay, we'd like to study the fish not at the edge of the tank. So we've got a big tank and a fish on here. But anyway, that's what we've got. So the fish will be here. They like to be at the bottom of the tank. As you can see, we take pictures from 3 cameras and superimpose images, and we're able to deduce the coordinates. So these are coordinates that are re-rendered so that they look nice. It's not the original video in a sense, but these are the tracked coordinates of the So we are able to track them. And the first question you might ask is, where are they? Well, as we saw from the photo, they're mainly at the bottom. We can start to look at static properties. So this is so again, inspired by Ornstein-Zernicke, we know from the pair correlation function about the interactions between the systems. Now, of course, that doesn't hold in an active system, but you can pretend it does, which is what we did. So this is a measure, the so-called attraction is a measure of the first peak of the pair correlation function, which will be evaluated in the passive system. And so that's what we mean by attraction here. We can also talk about nearest neighbor distance, and you see that these things are correlated. We can also look at their dynamical properties. This was the polarization that I mentioned previously, and we can see that the speed is highly correlated with the dynamic. So we can measure properties about the fish much as if they were active colloids. So we had some ideas for the states. Now this, of course, is a state where they're here, but then there might be a strong attraction between them. If, for example, they face danger, they might block if they're all going in this direction, and they might just be random. In fact, what we found is that the system changed its behaviour over time. So what's happening here is that this is the time fix, but this is the time to the experiments, the duration of the experiments, and they tended to move down into this form of the phase diagram, where they're going relatively slowly, not too much to fish liquid, but they sort of start over here, moving quickly together, and then be more disordered in time. Okay, so then one might imagine that one could induce an effective attraction between them by showing them danger, not a cat, a light, and so the lighter it is, the more they tend to condense, and the more the liquid at the bottom of the tank. Whereas when it's dark, they're more evenly distributed.
So did you try playing with different shaped tanks?
No, we've got a big one and a small one. They're not the same shape. So so the tank has a parabolic section, but that was the tank. Oh, hang on, no, sorry, that's not true. We confined them to 2Ds. We did some work where we confined them to 2Ds, so being about 2 centimeters high. So we did confine them, but we didn't particularly. Okay, so looking at them in the tank, what we're looking at the order parameters what we could measure the persistence length of their motion, okay, and then we could also measure the nearest neighbor distance. These things here are different data sets. O is an old fish, which is more than about 2 months old, and then Y are the young juvenile fish. There's no massive difference in the way we get them. The young fish are a bit quicker, but there's not critical to it. But what we found was that we could put a linear fit through the data if we look at the polarization order parameter and the persistence length and the nearest neighbor distance. The data seem to sit on a plane in this three-dimensional representation. And the BigSec model also describes a plane in this representation. We found that if we added inertia to the BigSec model, we got a rather good agreement between against the fish. So the line here is the BigSec model with inertia added to it. And what you see is that polarization as a function of reduced persistence length. And what I mean by that is that we're scaling the speed by the nearest neighbor distance. then this was then this gave a reasonably good agreement with the model plus inertia. Now, again, coming from a colloid background and using that as a way to approach fish, then with the colloids, they're synthetic, and we control their size and we can control their interactions to a very considerable extent. We can control the charge, we can have polarities to make them track one another, and put them in electric fields, as I showed you. So, is there any way to do something similar with fish? And the obvious thing is genetification, so this is not entirely coming from the colloid side, I must say. My collaborator, Chrissy Hammond, Bristol, was using the fish as a model system to understand osteoporosis. So the genetic modification, we didn't begin genetic modification in any sense. What's happening anyway, we took advantage of the genetically modified fish to do some experiments. Now, the reason she was using the fish to understand osteoporosis is that they're a relatively similar system to humans. Okay, there might be that might sound somewhat surprising, but at least in terms of what we can work on relatively easily, they're not too far away. So she was interested in osteoporosis, and these is some examples of what the effect of genetic is. So here's the spine of a wild-type unmodified zebrafish. And then what you see here, or indeed if we look more closely at it, is that the spine is really quite significantly altered by the genetic modification. It is the COL11A2 gene that is removed from the vision. So it's better expressed in the genetically modified fish. So one can then ask, well, the spines have changed. I showed pictures of the bones. The soft tissue has also changed extensively through this genetic modification, and the trajectories of the fish are also changed. This is an example. The wild type looks much more wiggly, whereas the mutant fish are tending to go in a much straighter line, and they're not turning. It's a little bit like this is driving on the highway, and this is driving in the middle of it. But anyway, although speeds aren't quite right. Okay, so where are they in the tank? Well, they're not in the same place. I said the wild type was in the tank. The mutants tend to be distributed actually slightly near at the top. But perhaps the thing I'd like to emphasize to you here is the speed. The wild types seem to have a bidispersed speed distribution. Sometimes they're basically stopped. But they also have a peak here where they're going very quickly at 200 kilometers a second. For the mutants, they just have a single distribution. And also we can think about measure how quickly the wild type fish turn at maybe a third of a second. For the mutants, it's much more like a second for them to turn. So this is entirely consistent with the fact that their spines are not great, right? It's difficult and probably painful for them to turn. So they represent that through swimming in a different way. This was looking at a single fish, but now if we look at 25 fish, then it's a similar story, perhaps less extreme, but indeed there is still these distributions of speed different, and similarly the wild type turn faster than do the mutants. So from that point of view, well, can we still describe the mutants with the big set model? The answer is yes. The answer, and even this parameter alpha that describes the amount of inertia we put into the big set model is the same. The only difference in the context of the model is where the mutant fish sits on the curve. So you see the wild type fish tend to be more at the bottom of the curve, the mutants are more distributed. So that's a sort of simple interpretation of how the fish are moving. And what I want to get to finish up with is thinking about other aspects of the zebrafish model system. So The zebrafish are described by using two order parameters. I talked about the polarization. The rotation order parameter here, what this is telling us about is are they doing what's called milling? So milling is where they sort of is what Bruce fish do. They sort of tend to move, swirl around in this sort of way. Schooling is when the polarization order parameters are large, they're all going in the same direction. And swarming is when they're just moving more randomly. And so there's this additional milling state that I'll introduce at this point. Now, geometrically, you have to have 3 fish to be able to observe milling. Okay? 2 fish do not allow one to see milling. So the minimum system size for this zebrafish behaviour is 3 fish. And if we look at 3 fish, what we see is that sometimes, so when it's purple here, it's their schooling, and when it's green here, they're milling, and then when both order parameters are small like this, they are swarming. So we can think about the 3 zebrafish systems and try to stay with state. And we might ask the question, well, what happens if we increase the number of fish? And if we do that, then I said that two fish cannot swarm because there's not enough of them here. But as we increase the number of fish from 3 onwards, we see the 3 states are occupied, and 4 fish are slightly more in the swarming state. And as we go up to 50 fish, in between there is a gradual transition to fewer and fewer in the schooling in milling states. What I mean is that there is no, as we make the system bigger and bigger, there's nothing like a phase transition. It's just gradually spending more and more of this time in the swarming state. Now, what we can also do is we can say, okay, so we could look at 3 fish in a group 50. That's what we're doing here. And if we look at the behavior of 3 fish in a group of 50, or 2 fish in a group of 50, or 4 fish in a group of 50, with these order parameters, it is almost indistinguishable from the isolated fish. And if, like me, when we're trying to compare fish to Leonard Jones atoms or colloids, this would seem quite odd. All right, so the organization of small groups of particles in isolation is quite different to the organization of small groups of particles in a densely packed system or in a large system. And yet the fish do not seem to care. What this means is that they're mainly interacting with their nearest 2 neighbors. Okay? So it's only what's governing their behavior is the 2 neighbors. So what I mean by 3 fish in 50 is 1 fish, its 2 neighbors, and then we're ignoring the rest of them. So in other words, the fact that that consideration is so similar to 3 isolated fish means the 47 further away fish don't matter. Okay, they're not really influencing what. So
in this, your groups of 3 or 4, are they exchanging partners? So
yes. They will exchange partners. Now, it turns out that doesn't matter because to calculate these order parameters, that's also convenient for another reason because we can't track them in time for very long. But we are to obtain the order parameters, we need the instantaneous velocity. So we take the instantaneous velocity of the fish and its 2, for example, 2 neighbors at one particular time, and then we don't but that's the only measurement that we make. So we're not, the change in partners doesn't actually influence the way that we measure. But yes, the rule they will change.
How do you get billing for this group? This group is the leftmost column.
So this one, the O, I don't have a good answer to that one. I will get back to it. Yes, I noticed that. What is going on here is that I think it's I know what the comment. I think it's a swarming state that we can't have, but too much. So what this leads us to is that the three is a crowd, right? So what I mean by that is that three fish is not different to many fish fundamentally, at least in what we can imagine. And but also the thing about that, then further away neighbours are a kind of fish soup, right? They don't really determine what's going on. Okay. The last thing I want to tell you about is the higher order correlations. Okay, so I started saying early on, I said in passive colloids we can study higher order correlation functions from the real space analysis, and these higher order correlation functions. One of the simplest is Kirkwood superposition. So Kirkwood superposition posits that the three-body spatial correlation function product in terms of three particles is the product of the three pairs. Okay, so one might want to inquire, does this hold in the fish? It works pretty well in passive systems, at least in the dilute case. It also works pretty well in the, I'm looking at the schooling state. In the fish, it works pretty well in the milling and swarming states, but in the schooling state, this is everything. So the three columns, this is the Kirkwood superposition, this is measuring three isolated fish, this is the three fish in a group of 50. Again, there's very little difference between these two columns. That is to say, the three fish do not care if they have 47 neighbours or not. But the Kirkwood superposition up here looks fairly similar if the fish are swimming side by side. Right? But if the fish are following each other, it's quite different. So in fact, so the Kirkwood superposition seems to break down in the case of the fish are following each other, whereas otherwise it's okay. So Kirkwood superposition works to some extent with a system of the zebra. Okay, so that pretty much brings me to the end of what I wanted to tell you. I talked about these new phases in the Quincke Rollers, and I've also talked about the active colloids in 3D, and I told you about the fish. That is to say, we've got the fish can be well described by the big set model with inertia, and there's also this three-state system that the three fish occupy, and that's fundamentally, at least in terms of what we can measure, it's fundamentally not different to the many fish system. And finally, a question we do have is, or going forward, is why is there an order transition in the Quincke Rollerdumbbells? Okay, we've got no explanation for that at all. Why is it? We know why, we think we know why dumbbells go in circles, always at the same radius, but we don't know why they decided to order, to go in a much more ordered trajectory at a certain distance. So with that, I'd like to say thank you very much for your attention.
Thanks for a rather, I would say, intensive summary of active systems. Any questions?
Yeah, I was wondering, in the with your fish, in the fish model with the I don't know how to read that, the inertia something model.
The inertial mixed model.
That's it, thanks. You have like they're like the older ones kind of clustering lower on the curve and the younger ones higher. And then in your other system where you had the osteoprophis fish, those were higher up the curve as well. So I was wondering if, are they like, yeah, what does that mean anything?
So do younger fish, I mean, it's the way they swim, so to the obvious. I mean, so we're measuring the swimming speed and the polarization at this reduced system. It's not, no, that's a very good point. We haven't thought about it at all. Thank you for raising it.
I was just wondering why, like, if it was
the young fish do look like they've been
yeah.
in that interpretation. In this reference.
Yeah, it seems like the wrong way around as well. If I imagined the older ones would be more like the old, like osteoporosis kind of ones, and
okay, so the fish tend to be more, the younger ones be a bit more nervous. When they get put in the tank, they're in their living tank, which is just normal fish tank, and then they get put in the observation tank. While they just don't know what's going on, and they get used to it. But in that while, when they don't know what's going on, they're sort of batting along quite quickly, and it may be that in that time through the analysis we did, it looks more like the mutant fish because they're not turning around.
Mm-hmm. Thank you.
So thank you for the point.
So staying with the zebrafish, you know the flow or the swimming patterns in the wild type and the modified ones. And you also know something about how the skeletal structure and maybe the flesh, so to speak, has changed as well. So in principle, you should be able to correlate mechanical changes to swimming patterns. Have you looked into that?
We've done it at the level of saying they don't like turning around as much, and that's pretty much it. I mean, they don't. Oh, sorry. We have confirmed. What we've done is we've done a. We've done a principal component analysis of their bend, and in short, so we can measure how much they bend. During the swimming, we imaged a two-dimensional system, and they were sort of wiggling like this, and we imaged how much they were bending. We made a principal component analysis of their shape, and I mean, yeah. So the upshot is that the mutant fish bend a lot less than do the environment type thing. And we have, in addition to these data, I showed these data because even I can look at a bone and go, oh, it's different. Whereas the soft tissue stuff requires lots of long words that I don't understand to say meaningful that. But they are certainly strongly affected. Even with the soft tissue between the bones are strongly affected. But to go from that to saying more than, well, they bend less, that's a sort of, well, yeah, I'm not quite sure how to make the jump.
Yeah, so I've got a question about the VTREC model with inertia, and using it to interpret your data, and I guess, and the fact that, I mean, because the VTREC model, of course, has collective behavior and transitions, and so it says that you can't separate. I mean, typically in the VTREC model, you can't separate a subgroup of particles from their environment. So is the claim then that the inertia somehow basically stops this collective behavior from emerging? Is that the
we don't see the ordering position of this, which is not in the phase that I've done. And I think the suppresses it, doesn't kill it.
Okay.
But it suppresses. So
you're saying that basically, so if I'm to understand it correctly, you're saying that you are in a regime when you are below the order to disorder transitional VTREC model, and the inertia just makes it actually makes it even less likely to plonk. So basically, yes. We don't. So okay, fine. So the argument is that you are. So now the argument normally with the order disorder transition is governed by some strength of the aligning interaction and noise. And so the claim would be here that the aligning interaction is weak, essentially. Okay.
Did you look at mixtures of wild type and mutant fish?
Don't know what we did. No, so it just no, we didn't. Sorry, that would have been a good idea, but no.
Biologists wouldn't care.
We might. I'm not. We might have struggled to distinguish because collectively they're different. But I mean, I showed those two trajectories, and yes, but of course these were chosen to look like it. Well, I don't know.
It may separate.
No, you're right. But I'm not completely sure we could have distinguished. but we can certainly, yes.
I mean, following up on that, you might imagine that a mutant fish, if it's influencing a nearby wild type, it will cause the wild type to have a more extended trajectory. So there could be some interesting interactions.
Okay, I've got a, okay. I'm gonna ask the previous question, but I'll wait on that.
The plot you showed for the polarization in the Witcheck model, you had at the lower axis the persistence. Can you show that? Yeah, and what is the
So,
area?
So we're sort of, if you like, we're scaling the persistence
By the nearest neighbor, but how is it with the size of or the curvature of the container? Does this also enter maybe? Or?
Well, the bookset model is not in that same kind.
No, but your fish
Yeah,
are.
what I'm saying is, I don't think it makes a huge difference. It would be nice to have a container that was big enough that didn't go to boils. So I think I'm just going to say we hope it doesn't play the things, but I think it's hard to test because the fish just swim towards.
So I can struggle. Your fish, are they point particles in your model or are they ellipsoids?
In the big set model of point particles, we have another model, which I'm going to tell you, I'm going to be really annoying and tell you that we have it, but not tell you about it. This thing here is a more complex model, it's a perception model, where they have, they can sort of see each other in certain, they interact in a certain spatial code around each fish, and they have attractions and repulsions. So this one is a substantially more complex model that we've developed, which gets the higher order correlation, which are not working. So this model, yeah, will have physical size for the fish, but not.
Okay, yeah, so the yeah, so I guess it was just a comment, and then there's you know people in the sort of ecology community, people like Ian Kazan, they have these very complicated models of fish schooling and flocking with lots of related to the brain and stuff like that. So I mean, so they look for me really, really complicated and very different from these. So do you have to say something about?
So the big set it gets the polarization and so on. This model actually does. So we took a cousin model, and our model was better than the cousin model in terms of the order parameters that we're looking at. So these order parameters here, and also the higher order spatial correlation functions. We tested our model because we started using the cousin model and decided it wasn't very good. We developed this. So that's where we are with it. But I appreciate he's done many. We just took a particular point.
Yeah. So I mean, so in some sense, do you have like a kind of opinion on these types of models? Because I find them very complicated. And
I think someone said as simple as possible, but no simpler.
Yeah.
So something like that. So the big set model is not going to describe this. But so we're using more complex model here, but at a minimum level. In fact, the reviewers told us to make, so this one we fixed the speed, and the reviewers from Cousins Community told us to make the speed vary in the model, and it made no difference. At the you had a plot with constant speed and plot with variable speed, and you couldn't tell which was which. So we're happy with this.
And finally, I'm going to ask a frivolous question, which is the student who did the experiments. Have they finished their PhD? And have they gone on to what are they doing now? And can you do more experiments? Are you doing more experiments?
We're not immediately doing more experiments. It's not possible. The experiments are not that hard to get, technically not that hard to get going. And crucially, the student is still responding to my emails.
Okay, so thanks, Paddy, for a very stimulating talk.